Hi students, welcome to my biology class. My name is Dr. Suja Agustin. In today's session, we are going to study about transcription. This is from your 12th standard syllabus, chapter molecular biology, right? Now, this part is highly important for your NEET examination as well as for your board examinations. For example, for your board examination, this transcription as such can come as a 5-mark essay question, right? Now when you look at your NEET examination, there are 3 steps when we discuss about transcription. So from each step, they can ask some questions. So when we are dealing with this session, that time I will update, right, which are the important NEET questions which are going to come from this side, normally for the NEET examination. So, transcription. Now what do you mean by transcription, right? You know about the central dogma of molecular biology, right? There we will study DNA undergoes replication. From here, mRNA is formed, right? Information which are present in a correct, this is replication, okay? DNA undergoes replication. Now the informations which are present in the DNA is rewrite, right? We will be able to rewrite, or the cell is rewriting into mRNA. From mRNA, so this step we will call it as transcription. Now from mRNA what will be formed? Protein. Now this step we will call it as translation. So this phenomenon we will call it as central dogma in molecular biology. Now when you look at this step, right? Transcription and translation, you have to always remember. Sequence is easy to remember. You know the alphabets, right, in English. So, DNA. D comes first. N comes after that only. Then comes protein. So, if you know the alphabet sequence, right, letters, then this is easy to remember. The same way, transcription, okay, up to trans, in both the cases it is same. Now C comes first. So DNA to mRNA will be transcription. Translation, right? S comes, right? After S it is L. So L comes after that. So you can easily remember mRNA to protein will be translation, right? Why I am telling such type of thing to remember? During NEET exam time, hardly we are getting 30 seconds to solve a biology question, right? As always I used to tell, the remaining time you have to use for physics, then only you can clear your NEET exam. So in 30 seconds, if you know these type of tips, right, then you can solve your question in the easiest way without any confusion, because whenever you are studying you will not get confused. The moment you will get a question paper and you are under exam pressure, adrenaline is there in your body, it started activated, that's it, done. Every sentence, whatever you will read in your entrance exam, you will get a doubt. So always remember this. Now you understood, okay? So just let me remove this. Okay, here's something I have written, no? I'm keeping it. Now, so you understood what is central dogma. In central dogma I told you, from the DNA, the informations are copied into mRNA. Or we can say, the method of copying information from DNA to RNA. Because we have 3 types of RNA, mRNA or messenger RNA, right? We have mRNA or messenger RNA, tRNA or transfer RNA. Then we have rRNA or ribosomal RNA, right? So that step we will call it as transcription. So when you think about transcription, first thing you have to understand is about the DNA, about its polarity. One strand is moving in 5' to 3' direction, the other strand is moving in 3' to 5' direction. So DNA has 2 strands, but when you create an mRNA, you cannot transcribe both the strands. Reason I will tell you. See here, if I have A, right, here it will be opposite to it T. Here if I have C, here I will have G. Now, if I one mRNA here and another mRNA here, both the mRNA will have different sequences, right? Now what will happen? Both these strands will be complementary. It will result in a double-stranded mRNA. This double-stranded mRNA cannot be, okay? This double-stranded mRNA cannot be processed by the ribosome because ribosome helps in the production of proteins and the information is present in mRNA. This is one reason. Got it? Now, if at all we are producing two mRNA, the information, the sequence present in them, it differs. Now, see for example here ACC is present here, TG, G was present. Okay, when they will form mRNA, right? mRNA also, what will happen? The sequence will be different, right? Two different mRNA will have two different sequences, which will result in two different type of protein, right? But we are going to produce only one type of protein. So once again, this will complicate the transcription machinery, right? And once again, it will complicate the translation machinery. So, is it clear? So normally, whenever we are thinking about transcription, always remember transcription happens only with one of the DNA. Now, which is that DNA? That is also important, right? So we have 5' to 3' and we have 3' to 5'. These are the two strands available in a double-stranded DNA. Now we have to remember, in biological system, the enzymes, correct, which enzyme we require? Here we require DNA-dependent RNA polymerase, which can process anything only in which direction? 5' to 3' direction. So that means if this strand we select, we have to synthesize in 3' to 5' direction. That is not possible, right? We can synthesize only in 5' to 3' direction. So now you understood, this will be the strand which will be selected, right? Based on this, so this was DNA, our DNA-dependent RNA polymerase. The DNA-dependent RNA polymerase is able to transcribe, right, only 5' to 3' direction. Because of that, 3' to 5' strand is going to act as the template. So this strand we will call it as the template. Okay, this strand we are calling it as a template. So what is the other name of the strand? This strand we will call it as the coding strand. Okay, so this strand, otherwise we will call it as non-coding strand. Now you will wonder why this strand 5' to 3' direction is called as coding strand. Because when you look at the sequence, see for example here if it was A, T, G, remember, and here it will be opposite to A, T, opposite to T, it will be A, and here it will be C, right? Now when you create an mRNA, okay, 5' to 3' direction, opposite to T, here it will be A, in place of A, in RNA which we have to use, right? Opposite to A, we need T, but T is not possible in RNA, we will get U, now here it will be G, right? Opposite to this, G will come. Now, when you compare these codes, just see, AUG is here, ATG is here, right? Except T, U is present. Otherwise, the codons which is present in which one? The newly synthesized, that is the primary transcript, will have the codon similar to that of the 5' to 3' strand. That's why we are calling it as coding strand, because the codons are similar here. Okay, this is clear. So this is the first point you have to remember for your NEET exam. Even when you are writing an essay, you should know this. We have a DNA. From that DNA, we are going to create the mRNA or copy the information into mRNA. That time, the 5' to 3' direction strand, we will call it as coding strand. Whereas the 3' to 5' strand, we will call it as the template strand, because it is going to act as a template. On that, we are going to create the mRNA. It is otherwise called as the non-coding strand. I hope this part is clear. Okay, we'll go go very slowly. This is the first part. Whenever you look into DNA, the entire part of DNA is not transcribed, right? There are some selected regions. There are some selected regions which is only getting transcribed. So those selected region, we can call it as a transcription unit, okay? The region which is getting transcribed, we will call it as a transcription unit. Okay, now when you draw a transcription unit, okay, we have three regions in the transcription unit. Okay, here we have 5' to 3', here we have 3' to 5', and in the first we will call it as the promoter region. The second we will call it as the structural gene. Okay, structural gene. The last part we will call it as the terminator. Okay, terminator. So a transcription unit consisting of a promoter region, then comes the structural genes. Okay, structural genes, which from there only we have to produce the mRNA. From that part we have to create the proteins. The information for proteins are present here only. Now the terminator region. Now what is the function of these three regions? Promoter region, okay, this is a region which is identified by the enzyme. Which enzyme? Okay, we will discuss about this enzyme little later. It's a DNA dependent RNA polymerase. I will tell you about the enzyme. Now structural genes, information is present here. Now the terminator region, right? Terminator region helps in the termination, the stopping of transcription. Okay, so this is about the transcription unit. Now we will think about the enzyme. The enzyme I told, okay. So when you think about transcription, we have two types, right? One, the transcription which is occurring in a eukaryotic cell, and transcription which is occurring in a prokaryotic cell. In today's session, I am going to discuss about the transcription which is occurring in prokaryote. Okay, so Escherichia coli you can take. Here you have to remember, when it comes to prokaryote, only one DNA dependent RNA polymerase is present, which will carry out the transcription of all the three RNAs, that is mRNA, tRNA, rRNA. Yes, that's an important main question. In the case of prokaryote, we have only one DNA dependent RNA polymerase which will carry out all transcription of all the three RNAs, mRNA, tRNA, rRNA. Okay, that is clear. Now we are going to discuss about the transcription in prokaryote. Escherichia coli I told, example, right? Bacteria. Now what do you mean by prokaryote? It's easy, right? Pro means primitive. Karyon means nucleus. So primitive organisms. That means they don't have a well-defined nucleus. So example, bacteria. Now, DNA dependent RNA polymerase. So never ever try to by heart anything in biology. DNA dependent RNA polymerase. First, why it is called DNA dependent? Because the template here is a DNA, that is 3' to 5' direction, correct? The DNA which has polarity 3' to 5' direction. On that DNA we are going to synthesize, based on that DNA we are going to synthesize one RNA. So that's why it is called DNA dependent, okay? RNA polymerase enzyme. So polymerase, an enzyme which is going to perform polymerization. That means it's going to make a polymer, right? Along nucleotide. Which type of nucleotide? Polynucleotide of RNA. So it is called as RNA polymerase. I hope the word meaning of DNA dependent RNA polymerase is clear. Now about this particular enzyme, we have 5 subunits, okay, in this enzyme. We have alpha, beta and beta dash. So we can call it as alpha to beta, beta dash. This part of the enzyme is called as core enzyme. Why it is called as core enzyme? Because the main enzyme which will perform polymerization, that is our core activity, right? Core activity, that is polymerization. Now there is a sigma factor. Then sigma factor attached to this enzyme. This part. Okay, 5 parts are there now. This we will call as the hollow enzyme. Okay. Now, sigma factor is required for initiation. So, I hope about this enzyme it is clear. This is DNA-dependent RNA polymerase. Now, we are going to our transcription. Transcription involves 3 steps. Okay. So, I am writing initiation, elongation and termination. These are the 3 steps in transcription. Now, when you look at each step, okay, for each step we require certain requirements, right? So, what are the requirements of transcription? First, already you know, we require DNA, which is having a polarity, right? Which is our DNA with coding strand and non-coding strand, right? Now we require DNA-dependent RNA polymerase enzyme. We require nucleotide triphosphates, right? We require, which are they? D. We require UTP, ATP, CTP, GTP. Don't worry about T, TP. TTP is not required. Reason, we are not going to synthesize DNA. Guys, we are going to synthesize RNA. So, UTP. So, the 4 different type of nucleotides we require. That already I told you. Yeah? We require some factors which will help in these steps. So, initiation step we require initiation factors. Elongation we require elongation factors. Okay. So, each step we require some factors which will help in steps. So, we require initiation factors, elongation factors, like that. Okay, that is also additional things which we require for transcription. Now, we are going to the first step, initiation. What do you mean by initiation? Initiation means, right, from the word meaning, we have to start this process. Which process? Transcription. Now, how we can start this process? Already in our body, there is a DNA which is available. Just see, there is a DNA which is available. This is the transcription unit. We have promoter, structural genes and terminator region. So, a transcription unit is already ready. Now, the enzyme has to come. Which is that enzyme? Yes. The enzyme is the DNA-dependent RNA polymerase. Now, the enzyme has come there. Okay. But how the enzyme will identify you? Have a long stretch of DNA that this is the part where I have to start the initiation. Right? So, how the enzyme will identify it. Okay. So, just imagine this is a promoter region. Okay. The transcription unit, when you look at this is 3' here, this is 5' here, right? This is a 5' region. Okay, in relation to the 5' region, we have to tell about which one? The promoter, okay, the sequence. So, there are 2 sequences which are present in the upstream. This is called as downstream. Okay, this is called as upstream. So, in this side, okay, this is the region, correct? This is the structural gene. So, here only we have to start our initiation. Who? Not me. DNA-dependent RNA polymerase has to start the initiation from here. That means from here, RNA has to be synthesized. mRNA has to be synthesized. Now, upstream, okay, around 10 from this is the starting point. Okay, this is starting point of transcription. From here, 10 nucleotides. If you see upstream, there is a sequence. Okay, of, we will call it as Tata box. The sequence is TATAAT. Okay, a 6 nucleotide base sequence is present. That we will call it as Tata box. Where it is present? In the promoter region, right? Upstream of the starting point of transcription, there is a Tata box. There is one more sequence that we will call it as -35 sequence. Okay, that means from the starting point of transcription, if you go upstream, -35, there is another sequence that we will call it as TTGACA. Okay, so these are the two sequence we will call it as. These are the two sequence we will call it as. What? These are the two sequence we will call it as the identifying sequences, right? The promoter region has two sequences. Recognition sequences are present. One is the data box and one is the, which one? TTGSA minus 31 sequence. Because from the starting point it is present upstream after 35 nucleotides, right? Now, data box is otherwise called as prib-now box. Okay, otherwise it is called as prib-now box. Yes, so you got two important neat question here, which is what is data box, which are the recognition sequence present in the promoter region, data box and minus 35 sequence. Now, data box, 6 nucleotide, it's TAT, AAT. Same way, minus 35, TT, GSA. Okay, so I hope this is clear. This is what happens in initiation. Okay, in initiation, what is happening? In initiation, okay, let me clean. In initiation, what is happening? In initiation, the enzyme, which is that enzyme? We had our DNA-dependent RNA polymerase. Okay, it's going to identify. How it will identify? With the help of sigma factor. So the coenzyme will take, coenzyme means alpha to beta beta dash. It will take the sigma factor. Together, it will go and identify the sequence. Okay, two sequences. So that's what we will call it as initiation. Once this initiation happened, the duty of sigma factor is over there, right? So what will happen? Sigma factor will move out. Again, it can go and activate another DNA-dependent RNA polymerase, right? So it's a cycle, it will continue like that. Now, after that, it is the next step that we will call it as elongation. So, what do you mean by elongation? I have to elongate. What I have to elongate? I have to make a polymer of mRNA. How I will make it? With the help of the coenzyme. Okay, the coenzyme is going to read the sequence. Which sequence? The sequence which is present in the 3' to 5' direction, right? And based on that, see for example, in 3' to 5' direction, one A was present. Opposite to that, what has to come? U has to come, right? Now what will be actually coming? UTP will be coming, right? Uridine triphosphate or uridylate, we will tell. Uridine will be coming there. Uridine triphosphate will be coming there. In that, we have 3 phosphate molecule, right? The pyrophosphate, that means 2 phosphate molecules will be broken down. Only UNP will be added here. Only monophosphate will be added here. Now, why we need this step? Because the energy which is coming from this 2 phosphate molecule need to be utilized. We need energy, right? Then only we can add 1 UMP, then we have to add next one. If it is GMP, like this we have to add it, right? For that energy is required. So like that in elongation, what is happening? With the help of elongation factors, the coenzyme alpha to beta beta dash, right? Alpha alpha, beta beta dash. This particular coenzyme will be reading the sequences, right? Which is present in the 3' to 5' DNA. Based on that, it will be start adding. In which direction? In the 5' to 3' direction. It will keep on adding the nucleotides, right? So that is what going to happen in which step? In the elongation step. That's why we are calling it as elongation. So here can you see elongation is happening. This is a newly formed RNA, right? So this is your core enzyme. As elongation proceeds, right? Can you see? We started from the 3' direction. It's going to end near the 5'. Almost it reached the 5' end. Once it reached that terminator region, okay? So next is a terminator. That means we have to synthesize only with what? We have to synthesize the structural gene, right? We have to code the information which is present in the structural gene into mRNA. Now it reached the termination region. Once it reaches the termination region, termination has to happen. Termination has to happen. That means we have to stop what? We have to stop transcription. Now how we can stop transcription? How we can stop this transcription? How we will be stopping the transcription? Easy. We have two methods here. One we will call it as row dependent. Second we will call it as row independent. There are two methods of termination. One is row dependent and second is row independent. These are the two methods of termination in transcription. Now, what is row dependent? That means we depend on a factor called row factor. So can you see here row factor? Row factor, either it will go and bind to the coenzyme. Once it binds to the coenzyme will move out of the DNA. This is one possibility. Second thing, it can transiently bind to the newly formed or newly synthesized mRNA. In that case also what will happen? The termination will occur. So this is the way row dependent termination will occur in transcription. Next method, it is row independent. Now what do you mean by row independent? When it comes to terminator region will have some palindrome sequences. So we have row dependent and row independent. Row independent means in the terminator region, okay, we have some palindromic sequences. Once the palindromic sequences are transcribed, it will result in the formation of some hair loops. Okay, once hair loops are formed, our enzyme cannot correct. There is a hair loop. Because of that, it cannot proceed further. So here what will happen? It will stop the transcription. Okay, so that we will call it as row independent. Okay, now how this is formed? It is easy. So we are going to create one palindromic sequence. So I'm taking the DNA 5' to 3' 3' to 5'. I'm going to take the DNA in 3' to 5' direction. We have C, C, G, 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 right? Again we have simple DNA sequence. Opposed to this, I am going to create an mRNA in 5' to 3' direction. So it will be G, 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 C, 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 G, 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 C, C, C. Right? Now, when I am writing this mRNA, okay, like this, just imagine G, 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 C, C, C. This part will loop out. Okay, loop will be formed. And here G, 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 C, C, C will come. Right? This is the 3' direction. So, this type of a sequence, a palindromic sequence, when it is present, what will happen? It will result in the formation of hair loop. Hair loops are formed like this only. The newly formed mRNA will create palindromes. The newly formed mRNA will create hair loops. Once hair loops are formed, what will happen? The termination will occur. So, with this, we completed transcription. So, what are the key takeaways? First thing, you have to understand about the transcription, the definition of transcription, right? Then you have to think about transcription when you think about the board level. It can come as a 5-mark essay question, right? But when it comes to NEET level, we have n number of questions, right? So transcription, we had 3 steps: initiation, elongation and termination, right? Initiation, we were discussing about prokaryotes. So only one DNA dependent RNA polymerase is there. It transcribes all the 3 RNAs, tRNA, rRNA, mRNA. That is first thing, first key takeaway. Then after that, when you look at the enzyme, we have subunits, right? I told you about alpha, alpha, beta, beta dash. Then we discuss about rho factor, sigma factor. Then we studied about elongation, right? So all these factors, right? Row dependent and row independent termination. All these things are important for your NEET exam, right? Now you have to remember about data box. You have to remember about -35 sequence. What is the sequence there? TTGACA or TATA, right? TAT. So data box that you have to think. Okay. So Thanks a lot for listening to my class. Thank you. We will see in another class.